So I'm Wim Thielemans from the Sustainable Materials Lab at KU Leuven, and this is an isothermal titration calorimeter. So this is a large device which keeps the temperature constant to about 10 to the minus 5 Kelvin, so 0 is 0.00001 degree exact. And we use it to look at or to measure interactions between nanoparticles suspended in a solvent and another species. And that can be an ion, that can be a protein, that can be other nanoparticles or polymers, for example. In the AAA codes project, we use it to look at the interactions of cellulose with antibacterial proteins or compounds, antifungal compounds that might absorb into cellulose, so we can actually measure how strongly they absorb onto the nanocellulose to determine whether the binding just by absorption is strong enough or whether or not we need to have a chemical linkage. So how does this work? It's a calorimeter, so you have two measuring cells. On one hand you have a reference cell which looks like this, and that you're going to add either a metal sample holder or a glass sample holder. And in this sample holder, you basically have your nanoparticles in the solvent that you want. We do it in water for these experiments. And then you close it, you put it on there, and you lower it very slowly into the calorimeter to make sure that the temperature stays stable. And then you have a second cell, and this is your measurement cell. And there you're going to use exactly the same sample holder with exactly the same suspension of the nanocellulose in water. And then what you can do is, after you have lowered both of them in there, you have a syringe with your protein or your antibacterial or antifungal compound that you want to interact with the cellulose in solution, and you're going to inject it via a very long needle that goes all the way down into the sample cell. So you see it here. This is a sample holder outside of the calorimeter, and you see here a needle that goes all the way down. So that's a hollow one, and in there you can insert the actual needle that goes all the way down. And then you start injecting very, very small amounts, and we can measure the interaction energy very, very accurately between what we inject and the actual nanoparticles in there.